Welcome children. In this video I will be dealing with some high order thinking questions, that is reasoning type questions. The first reasoning comes with respect to the genetic material. We know that most of the organisms have DNA as a genetic material, though RNA is also serving as genetic material. Most of the organisms have DNA as a genetic material. Only few organisms have RNA as a genetic material. For example, the tobacco mosaic virus, then the HIV virus, and only few retroviruses have genetic material as RNA, but most of the organism has DNA as a genetic material. So we are going to analyze why. Then the second why is with respect to DNA replication. We saw that the DNA replicates and it opens by a small split at one end. It's a very small opening. Why? Why can't it separate its entire length? And the third why is with regard to transcription. We said only one of the strands serve as a template. What if two strands serve as a template for transcription? Okay, so let's start off with the first why question. DNA is the predominant genetic material. It is on page 103. For a molecule to be a genetic material, it should satisfy some criteria. So some criteria we are going to analyze now step by step. The first criteria it should fulfill is that it should be capable of replication, that is creating its own replica. Second criteria, it should be stable, that is both structurally and chemically. Third criteria, it should have the scope of slow mutation or slow changes via mutations. Mutations can cause the changes very fast and very slow, but here what is required? Slow changes by mutation. And the fourth criteria, it should be able to express in the form of Mendelian characters. So now we are going to analyze each character and see the three biomolecules. The three biomolecules that are going to contest this race are DNA, RNA and proteins. These are the three major biomolecules. Okay. Now, first criteria, it should be capable of replication. When we look into this criteria, DNA and RNA stands a chance. Why? Because of the rule of base pairing and complementarity. Both DNA and RNA having bases which forms complementary base pairing. Okay, in DNA it is ATGC, whereas in RNA it is AUGC. So in both these molecules there is chance of replication. They replicate because of this particular property, rule of base pairing and complementarity, which is not there in proteins. So henceforth I am going to rule out proteins, and proteins we are not going to consider proteins again. So it's again between DNA versus RNA. So in this, the first criteria, it is successfully satisfied by both DNA and RNA. Second criteria, it should be structurally and chemically stable. In this context, we know which is more structurally and chemically stable. It is the DNA. Okay, we already know, but now we can, the next slide I am going to see the reasons why DNA is more stable than RNA. The reasons for the stability in DNA. It has two strands. It is double stranded, whereas RNA is single stranded. Second reason, there is a base called thymine, whereas in RNA it is uracil. Thymine confers stability to DNA. Third reason, as proposed by those two scientists who found out the structure, the real structure of DNA, Watson and Crick, they said that stacked nature of the base pairs. We have learnt in the double helix structure, the stacked nature of the base pairs confers stability to DNA, whereas here there is no stacking of base pairs because it is single stranded. So there is no point of telling about RNA. And fourth is with respect to the chemical structure. We know in both DNA and RNA the nucleotide comprises three components. The three components are base, sugar, phosphate. Now we will be analyzing the sugar of DNA and sugar of RNA. Now the sugar of DNA is called deoxyribose sugar and the sugar of RNA is called just ribose sugar. Now I am going to draw the chemical structure of deoxyribose sugar. Here this oxygen, here this carbon, first carbon, second carbon, third carbon, fourth carbon and fifth carbon is here. Similar structure I am going to draw with another color for the ribose sugar of RNA. Same structure. I can just copy paste. So here this oxygen, first carbon, second carbon, third carbon, fourth carbon and the fifth carbon. Now the difference is going to come here. I am going to add the hydroxyl group, OH group. In DNA, in the deoxyribose sugar, hydroxyl is connected to the first, third and fifth carbons, whereas in RNA there is an additional hydroxyl group, one at the first position, second position and third and fifth position, and the hydroxyl group at the second position is only making it very reactive. So, I am going to add the hydroxyl group. This is first, second, third, fourth carbon and fifth carbon. So, here we have one hydroxyl group at 1, at 3 and at 5. Similarly, taking the ribose sugar of RNA, we have at 1, at 2, at 3 and at 5. So, what makes DNA more stable? The hydroxyl group that is lacking in the second position. It is not present in the second position of the carbon atom. Therefore, DNA is more stable than RNA. Or you can talk in terms of RNA. The hydroxyl group that is attached to the second carbon compound, this one, second carbon compound makes it very reactive. So, we say this 2' OH is responsible for making the RNA very reactive, or this side we can say lacking of the 2' OH. This is not present, therefore it is very stable. So, once again the reasons why DNA is more stable than RNA, the first reason is the 2 strands in the DNA makes it more stable, whereas RNA has a single strand. Second, the base thymine is present in DNA in the place of uracil in RNA. Then stacked nature of the base pairs in DNA, there we can't say the stacked nature because it is not double stranded. And lastly, it lacks, the DNA lacks an additional hydroxyl group at the 2 carbon compound of its sugar. Okay. But in RNA there is an additional hydroxyl group at the 2 carbon or 2' of the ribose sugar. So, these are the 4 reasons why DNA is more stable than RNA. In this context, we can say DNA has a better chance to be a genetic material. It is fulfilling that criteria. So, getting back, we dealt with the second criteria. Now, the third criteria. Now, we understood that DNA is more stable and RNA is very reactive. Opposite of stability, very reactive. So, in the third criteria, automatically we can answer which is having slow chances or slow change, DNA. DNA because it is very stable, it mutates but very slowly. So, here also we can see DNA has an upper hand because it is very stable, it mutates very slowly. It should mutate but very slowly. On the other hand, RNA is very reactive and mutate fast. So, in this case I will rule out RNA. So, better is DNA as a genetic material. And the last property or the criteria to be fulfilled is which expresses itself. Okay. DNA and RNA. Now, by the time we have learned protein synthesis and we know which of the two expresses fast. Talking about DNA depends on RNA for the entire process of protein synthesis. For example, this DNA depends upon mRNA, then it depends upon tRNA, and it also depends upon RNA. So all these raw materials DNA depends on it. Okay, we know that DNA, from the DNA mRNA is synthesized, and from the mRNA we see proteins are synthesized, and this mRNA, for it to get coded into proteins, it requires the tRNA, help of tRNA. So mRNA is needed, tRNA is needed, and not only that, to start off translation we know we require ribosomes, and to prepare ribosomes rRNA is needed to synthesize ribosomes. So we can see DNA depends on RNA for it to be expressed as proteins. It depends upon mRNA, it depends on tRNA for transfer of the correct amino acid, and it depends on the rRNA for synthesizing the ribosomal units to initiate translation. Whereas RNA, so we can start off with RNA. From here only I am starting. But look at RNA. RNA, is it depending back on the DNA for proteins? No, from here it starts off. So I am redrawing over it. This mRNA depends on its another species of RNA called tRNA, and it depends upon another species of its own, that's rRNA, to synthesize proteins. But it's not depending on the DNA, not depending on DNA. So RNA not depending on DNA for transcription or translation, or we can say for protein synthesis. Very independent. With the three strains mRNA, tRNA, RNA, proteins are synthesized. Okay, whereas DNA has to depend on mRNA, tRNA and rRNA for it to get expressed. So the first why is answered. Next why. Question, no, before going to the next why question is, from this what we can infer, which was the first genetic material to be evolved? It is RNA or DNA? Yes, it is the RNA that was the first genetic material to be evolved. There are many evidences to prove this. So it is from the RNA that the DNA came. So the first genetic material was RNA, and later only DNA was evolved. The first reason to prove it, life processes evolve around the RNA. Like say many life processes depends on RNA, metabolism, translation, splicing, etc., all depends on RNA. Second reason to say that RNA was the first to be evolved is some important biochemical reactions in the living systems are catalyzed by RNA and not by proteins as enzymes. We know most of the enzymes are proteins, but in some important biochemical reactions, not proteins are used, RNA are used as enzymes. And lastly, RNA is very reactive and unstable. So it will not remain for long on earth. It will get evolved or mutated, and it became DNA with a lot of chemical modifications. What were the modifications that occurred to RNA to become the DNA? So RNA has become the DNA. So what and all it has got modified? Single stranded became double stranded. Then in the place of uracil, thymine came, and automatically with all these changes, what happened? The stable component DNA was evolved from RNA. So this slide proves that the first genetic material amongst DNA and RNA, it is the RNA to be evolved. These are the three reasons or evidences supporting that RNA is the genetic material, not DNA, the first genetic material. However, going to the previous slide, when we compare these two, DNA and RNA, we can say DNA serves as the storage for genetic information, and RNA can serve for transmission or transfer of genetic information. So finally, when we make a comparison and contrast between DNA and RNA fulfilling the criteria of a genetic material, we can wind up with this very important statement, which once again I am highlighting. Because of the stability we saw in many cases, many of the criteria we saw, DNA is more stable. Because of the stability, DNA can serve for storage of genetic information, and RNA, because it is very reactive, labile and easily degradable, it can well be suited for transfer of genetic information. So finally we wrap up with the conclusion that both are genetic material, but better for storage of genetic information DNA, and for transfer of genetic information RNA. Now we will move on to the second why. The second why question or reasoning question is with respect to DNA replication. You are familiar with this. This is a replication fork. Now the question here, we started off replication by saying that it is small opening, very small opening at one end, which is brought about by the enzyme helicase. We have learnt under DNA replication. Now a question I am asking, why can't that helicase unwinds and divides or separate the two DNA right across the entire strand like a ladder? See, this is the DNA, why can't it separate like this? But instead it is like this. The separation is like this. Okay, there is only a small opening at one end brought about by helicase from a point called ori or origin of replication. Now the question is, why can't it be at one stretch? Why can't it separate into two separate strands? Or why the DNA cannot be separated in its entire length completely? Why can't it be separated? Like in PCR we have learned, in the procedure of PCR, the first step is application of heat so that the two strands of DNA separate. There the application of heat, very, very high temperature, high energy is used to separate. So the answer to this question is due to high energy requirement. So very simple, the answer is why it opens like at a very small opening at one end, and later it progresses. It does not, it finally progresses, but slowly, not at one stretch it separates. It opens up at one end due to helicase, and slowly it unwinds and opens up, and finally we get both the strands separated, which serves as a template for new strand. But why it starts off with a replication fork? Why it opens up at an end at a small opening? The reason is to separate it through its entire length, a lot of energy is required, and that we have seen in that PCR which is done by humans. For it's a test for getting copies. There the one stretch, the two strands of DNA are separated with a lot of energy, and energy in the form of heat. So much energy is produced or provided so that it gets separated. So you got the answer to that. It's very simple, due to high energy requirements. And the last why question, that is with respect to transcription. We saw in transcription the DNA of the DNA, only one of the strand, and that is the strand running the 3' 5' direction, will form the mRNA. So the strand which is running the 3' 5' direction will form the mRNA which is running in the 5' 3' direction. And we know the answer to this, why only one of the strands serves as the template for transcription, is because of the enzyme RNA polymerase. This enzyme functions in the 5' 3' direction. Because it functions in the 5' 3' direction, the template which is running in the 3' 5' direction is only serving as a template, not both the strands. Now I'm telling you of a condition, both the strands serves as template. Okay, so now I'm taking both the strands of the DNA serving as template. So let this be one strand, this is the second strand. So irrespective of the condition of RNA polymerase is not having such a condition that it functions. Assume, this is assumption, assume that it can function in any direction with not taking into consideration the orientation. It functions in a 5' 3' direction for the time being, assume. Okay, it can function in any direction. So let me take two strands. One strand is running the 5 prime 3 prime direction, the other strand running the 3 prime 5 prime direction. What happens if both the strands serves as template for transcription and translation? So just for example, I am giving the basis AAA, GGG, CCC, TTT. If these are the bases, definitely the basis of the other strand will be complementary. For the second strand, complementary to AAA, it will be TTT, complementary to GGG, it will be CCC, complementary to CCC, it will be GGG, and complementary to TTT, it's AAA. We know it very well, and I gave it in triplets so that it's easy, of the same alphabet for you to identify the codons. Now, so just imagine this is undergoing transcription, this is also undergoing transcription. Okay, so I am going to bring the transcript adjacent here itself. So 5 primes transcript will be, for AAA it will be UUU, for GG it will be CCC, for CC it will be GGG, and for TT it will be AAA. So this, what is indicated in violet, is the transcript of the strand running the 5 prime 3 prime direction. Now I am going to give the transcript of the strand running the 3 prime 5 prime direction with another colors, green. Okay, so here TTT it will be AAA, CCC it will be GGG, GGG it will be CCC, and AAA it will be UUU. Now, what will happen? The first foremost situation, what will happen? Both the DNA strand serves as a template for transcription. Yes, transcription took place. We got the violet strand from 5 prime 3 prime direction. We got the green strand from the 3 prime 5 prime direction. But look at the two strands formed. They are complementary to each other. Immediately hydrogen bonds will be formed between the two. Okay, between UNA, UNA, UNA. GNC, 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 then CNG, 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 and ANU and ANU. So what will happen? Will further step take place, translation occur? No, it will not take place, because the two strands that were formed by the two mRNA strands formed from the two templates are complementary to each other. Therefore, there is a tendency to form hydrogen bonds. And what is formed as a result? A double-stranded RNA is formed. So, is transcription a useful process here? No, it becomes a futile process, a useless process. It is not going to get translated into proteins, because the two mRNAs being complementary to each other formed a double-stranded RNA. So, it is not going to further undertake the next step, translation. So, one drawback is, the first drawback is a dsRNA is synthesized. Therefore, translation will not take place. Now, the second situation, what will happen? Now, the second situation, if both the strand is acting as a template, okay, we can go ahead. Here also we can see an mRNA synthesized, here also an mRNA synthesized. Okay. So, we know the basis of these are UUU, CCC, GGG, AAA. So, this is the basis complementary to the strand running in the 5 prime 3 prime direction. So, this strand, mRNA strand formed is running the 3 prime 5 prime direction. Now, the mRNA strand produced by the second strand, which is running in the 3 prime 5 prime direction. Ready? The bases that are going to be formed here in this mRNA, which is complementary to this. It will be AAA, for CCC it will be GGG, and for GG it will be CCC, and for AA it's UUU. Now, next step, they are not going to form dsRNA, we block that. Okay, we are all assuming what will happen. Next step, let it allow protein synthesis. Now, UUU, the proteins that is formed, if you refer page 112 of the genetic code, that is dealing with the genetic code, like the Clark's table, it should be referred, but not in your syllabus. I am just saying for U, which is the amino acid phenylalanine. For C, it is proline. For G, it is glycine. And for A, it is lysine. So look at the amino acid. The first protein synthesized will have amino acids phenylalanine, proline, glycine and lysine. Now analyze the second protein that is synthesized from this strand. Okay. For A, it is lysine. For G, it is glycine. For C, it is proline. And for U, it is phenylalanine. Now look at the two proteins. All of you focus at the two proteins. The proteins produced by one strand which is running in the 5 prime 3 prime direction, and the protein synthesized by the second strand, the strand which is running in the 3 prime 5 prime direction. Look at the sequence of the amino acids. Look here, in the first protein phenylalanine is first, but in the second protein where is phenylalanine? Fourth position. Then the first protein proline is in the second position, where is proline here? In the third position. Then glycine in the first protein is in the third position, but here glycine is in the second position. Lysine in the first protein is in the fourth position, but look at lysine in the second protein, it's in the first position. So what we can infer is, if at all its proteins are coded, what will happen? The second drawback, we get two sets of proteins with totally different sequence of amino acids, same amino acids. Look at the two proteins having the same amino acid, phenylalanine, proline, glycine and lysine. But the way here it is arranged in one order and it is shuffled and arranged in another order, reverse order in the second protein. So here we can see two sets of proteins are synthesized with a different sequence of the amino acid, and this makes the entire process complicated. The genetic information transfer machinery gets complicated. Now during this process of protein synthesis, there is a lot of complication. tRNA has to do roles to translate that mRNA and also to translate this mRNA. So there is lots of complication, though it was easily, I made two proteins using the board and the pen, but it's very difficult because the tRNA has to do the role. In this place proline has to come second position, here the very same tRNA has to put proline in the third position, it has to do multitasking. Okay, so the second drawback is proteins. Two sets of proteins are synthesized with different sequence of amino acid. Therefore, that genetic information transfer machinery becomes complicated. So who is doing the transfer here? It is the tRNA. The complication comes for the tRNA molecule. It is doing multitasking. See, tRNA has the problem. tRNA is specific for an amino acid. Okay, so we know for each amino acid a specific tRNA. So for example, if this is the tRNA for phenylalanine, it has to fix phenylalanine in the first position here and phenylalanine in the fourth position here. So it should be available on two strands and so difficult multitasking. So it is going to hamper the transfer machinery which is involved in protein synthesis. So once again I conclude with the drawbacks if both the strands of DNA are transcribed. The first drawback is dsRNA is synthesized. Okay, being complementary, the two mRNA being dsRNA is synthesized, and therefore will translation occur? Translation will not occur. So transcription therefore has become a futile process. The second drawback is two sets of proteins will be produced, and how will these proteins be? With different sequence of amino acids. Same amino acid, but the sequence is different, which means it gives extra burden for our tRNA. Same tRNA to fix the same amino acid. So multitasking for the tRNA. So therefore I gave it in a short form, which is very, very important. The genetic information transfer machinery becomes complicated. It becomes complicated for our tRNA. So with this I wrap up with the very important reasoning question of this chapter, molecular basis of inheritance. Thank you.